Let's start with a demonstration. This block is made of agar, a seaweed extract, a bit of the base sodium hydroxide, and a pH indicator. This pH indicator, phenolphthalein, is this very nice fuchsia color when it's exposed to a base, and it turns clear when the pH turns acidic. I've cut cubes of this agar into 4 different sizes. The biggest is 3 centimeters on a side, the others are 2, 1, and 0.5 centimeters on a side. I'm going to put these cubes into a dish of vinegar, a weak acid. Watch what happens. In 6 minutes, the tiny cube has lost its fuchsia color and turned completely white. The other cubes still have some fuchsia inside, and the percentage that's still fuchsia goes up with the size of each cube. In other words, vinegar diffused into 100% of the smallest cube's volume, while it reached only 19% of the largest cube's volume. Let's get our heads around what's really going on. Diffusion is the movement of molecules in a fluid, a gas, or a liquid from where they're more concentrated or bunched up to where they're more spread out. It's how this drop of food coloring is spreading throughout this flask. You don't have to stir or add energy. The random movement of the food coloring molecules will lead them over time to move apart from one another. When we put our agar cubes in the vinegar is more concentrated outside the cubes than it is inside. So because there's nothing from stopping the vinegar from moving in, the vinegar diffuses from where it is, outside the cubes, to where it isn't, inside the cubes. Cells, to stay alive, have to exchange materials with their environment. For most cells, that means that things like oxygen and food need to go in, and carbon dioxide and other wastes need to come out. That exchange is largely by diffusion. If cells were too big, then diffusion wouldn't work in terms of reaching the entire volume of the cell. Just pretend that this smallest agar cube was a cell, and that the vinegar molecules were oxygen. This cell is doing fine. Oxygen is getting all the way deep into the cell. Things are diffusing in and out. This cell is well adapted to exchange substances with its environment through diffusion. But if these larger cubes were also cells, they'd be having a hard time. Whatever needs to get in isn't getting in very far. A lot of this poor cell would be suffocating and filling up with waste that can't diffuse out.